we are ready to welcome the man and the legend, Alex Zhavoronkov and Dominika Wilczok, his research and not only partner. Alex is the legend, an absolute legend in longevity field. I would also call him my mentor, the man who I owe much to actually my career in longevity field. Alex is the founder and CEO of In Silico Medicine, the juggernaut in AI for drug discovery, and one of the leading companies. Oh, thanks, Victoria. Now you can see me. One of the juggernauts in AI for drug discovery, and he's doing RDD and many, many other things which move our industry a lot. Dominika is AI in longevity activist, and she is young but has accomplished a lot of things. So welcome, Alex Zhavoronkov and Dominika.
Well, first of all, thank you for such a generous introduction, and very pleased to be with you. Today we will be presenting together with Dominika, who is my longevity partner and longevity fiance, so to speak. I know that many of you are interested in starting something in longevity, or you are already doing something in longevity. So I hope that our presentation today will be helpful to navigate your pathway in this field. We'll start with a set of slides, and then we will open the floor for questions. Please feel free to also text those questions in the chat. We have access to Zoom. We are actually not far from each other with Dominika, but I got pretty terribly sick on my flight from the UAE, and today I will still present, but just mind that I have a form of bronchitis, which is not great for longevity, but that happens when you travel. So I'll present on AI for drug discovery. So today we're gonna cover three main topics: AI, robotics, drug discovery, and Dominika is gonna talk a little bit about cryo. Because with Dominika, we are kind of like Rick and Morty. So we are very complementary, and she joined this field when she was 14. And we met at the very prestigious aging research conference where she was presenting a poster with a mirror in it. So very dedicated to aging research, where you could annotate your face yourself and then read a poster and look for features that give out your age. And then we again met at the Nature conference on AI in healthcare, where she presented on multi-agent systems. So very capable young researcher, and together we're 1 + 1 = 10, hopefully. So we refer to ourselves as ZW, maybe WZ. Because she is now kind of spearheading many of the frontier projects. And a little bit about myself. So Alexei introduced myself, but three things that I am very happy about, so not proud of, but happy, is that I contributed multiple articles on aging research in peer-reviewed journals. Also in AI and generative adversarial networks, and reinforcement learning for small molecule drug design, for biomedical data analysis, and all kinds of simulations and generations. I co-founded ARDD conference, founded ARDD conference, but today it's managed by Morten Scheibe-Knudsen. And I made the longevity pledge, where I kind of pledged not to get married or have kids, but then I met Dominika, and now we are thinking about how to rework this pledge for greater good. And today I'm going to talk about my favorite topic: how can generative AI and robotics help us fight aging? And one lesson I want to convey, something that you definitely need to understand, something that took me a while. I am in aging research for 20 plus years, and before that built a pretty successful career in IT doing GPU. So before GPU was used for AI, or before it became so efficient at it. And when I entered the field, I thought that if you do public advocacy, governments will move and start funding longevity, or billionaires will crank up and open their pockets and start pouring the money into longevity. And I actually did pretty well writing the book, doing a lot of talks, 2009, 2010, 2011, all the way to 2014 when I started in Silico. Talked to countless high net worth individuals, joined multiple nonprofits, established nonprofits, and realized that whatever you do, you need to prove the concept. You need to demonstrate that something is really working, and not in just mice, but in humans. And you need to build a sustainable business model. If you don't have a sustainable business model that can last for a couple decades, preferably longer, and fund your research in aging and longevity, most likely you are going to die as a company and possibly as an individual. And one of the reasons for that is because, well, first of all, if you look back to 1970s, 1980s, 1990s, every decade you see a major wave of interest in longevity biotechnology. You've got companies like Life Extension, founded by Bill Falloon and many other people who came before him, contributed, and they were looking at not only supplements but other things, and then they kind of had to figure this model. But most of the, like all of the longevity companies that I've looked at over the years, they failed. And think about Geron in the 1990s that was going after telomerase. By the way, telomerase is getting hot again. They had to repurpose to cancer. You've got Elixir that raised money. You've got many others over time. Michael West is one of the true pioneers of longevity biotechnology, so to speak. There was a book called Merchants of Immortality that I highly recommend reading. And whatever you do, people thought about something similar, and also tried to align with the billionaires. Some of them did it successfully, but so far, no success. And one of the reasons for that is that even if you look at something like drug discovery, which can probably push us by 30, 40%, maybe 50%, if we are really good at the current traditional paradigm of delivering new drugs, and not only eliminate the diseases, but also figure out how to improve the human body with certain interventions. But if you look at the traditional drug discovery and development paradigm, the cost of developing one drug ranges from $2.8 to $6.1 billion. It takes about 12 years from the time you already know the mechanism of disease and the biological target to the time it gets approved, the drug gets approved. For example, the fancy GLP-1 drugs that are super fashionable today and probably are longevity therapeutics in a way, were discovered around 1980s as a target. Then in early 2000s, people started prioritizing it as a target and started looking for antibodies or small molecules. Now people have, drug companies have succeeded developing antibody therapies for GLP-1 and some other targets. And it's a multi-billion-dollar business. Over a decade, it might be $1 trillion business. And now those same companies that pioneer GLP-1s are looking at aging and longevity and prioritizing aging research for their drug discovery and development, which is great. So that shows you that some success, even with pharmaceutical companies not being focused on aging, if you get a drug on the market that addresses a biological condition that is chronic and yields significant profits, will result in more money in aging research. But you need to figure out ways to develop your drugs in the most credible, sustainable manner that generates revenue, attracts investment, and is not just a flashy idea that will perish very rapidly. So I decided to dedicate my life to improving this process. And Dominika is actually looking at more frontier technologies right now. So our industry has one huge problem in aging research. And there are so many ways to measure aging. Lots of aging clocks. I've published over 20. And there is only a handful of drugs that are proven to extend life in mice. And there are no drugs that are proven to extend life in healthy humans. So right now, there are, of course, you can argue with that, right? And say, okay, metformin, all those metabolic regulators, and you can extend healthy productive life in some ways, maybe by a few months with some additional medications. But in healthy humans, very, very difficult to make this inference, even when it comes to rapamycin, the drug I really respect, made by nature, but then reinvented by human ingenuity. And we still don't understand how rapamycin works. So we don't know what to take right now, right? Even experts in longevity struggle to figure out what works and what doesn't, because none of those drugs were originally invented as longevity drugs. So how do we figure out how to create drugs that are true aging drugs that have the commercial capability, commercial potential for investors and pharma companies to invest in those drugs and can be used in combination with aging biomarkers in order to prove that they actually reverse some of the aging clocks or extend healthy productive life or avoid getting certain diseases. So why do we have this problem currently? As I've explained, the cost of developing a drug could range from $2.8 to $6.1 billion, could be even more. The cost of developing an aging clock is close to nothing. So anybody here can use ChatGPT nowadays to replicate some of our work that we've done in 2016, developing deep aging clocks on blood biomarker data. But what are you going to do with those clocks, right? You can measure some aging processes, but you cannot really derive valuable therapeutic insights from those up until recently. So now I'm going to switch to how can we use AI and robotics to actually intelligently identify some of those longevity interventions that may address a certain disease or biological conditions and could be measured by an aging clock. And ideally, those therapeutic targets and drugs would come out of aging clocks. So you would use an aging clock to discover them, develop for a disease, and then use an aging clock to test whether they work in a disease and in aging. So how can AI help in drug discovery? So here are some benchmarks for any one of you to take home if you are thinking about discovering a drug, right? And I can tell you that if you raised something around this much, you probably have enough money to get to, let's say, 3 or 4 preclinical candidates before you run out of money and need to fundraise, if you are really good at it, right? I think that if you are developing a platform, you might raise that much and still not get a single preclinical candidate. But if you have a platform, I think that you need to demonstrate that you have 3 or 4. So since 2019, when we raised our first big round, we started doing our own drug discovery and managed to get 20 preclinical candidates nominated. A preclinical candidate is a drug that is one step away from human clinical trials. So what you need to demonstrate is GLP safety, so a formality. Our preclinical candidate package usually includes 28-day non-GLP toxicity. So we very rarely, or so far never, failed in GLP toxicity studies. So it's usually kind of a formality, but it takes you about 9 months to a year to get through GLP tox, sometimes faster, and get IND approval. 9 reached IND stage, so we got IND clear from the regulators. 2 are now in phase 2 human clinical trials, passing phase 1 safety. Now it's efficacy. 1 completed phase 2A, safety and dose-dependent efficacy. So look at this program very carefully if you want to benchmark your own programs, either with a small molecule or biologics. Biologics sometimes are even faster. Our shortest time to preclinical candidate was 9 months, and average time to preclinical candidate is now 13 months for moderately novel targets. So that's very important kind of benchmarks. This is how in silico looks like. So we have LLMs orchestrating a large number of models. We have over 700 models that have been proven to work. They've been validated using experiments, either on the chemistry side or on target discovery side, either on a generative side or reinforcement learning side. And we also have now close to 2,000 agents. By the end of this year, we'll have over 2,000 agents that perform certain human tasks, right? Data annotation, data curation, running certain models, running robotic systems. We also have software on the market, so with a pretty substantial community. Now, 10 of the top 20 pharmaceutical companies would be using our tools. But this is a very small market, by the way. So if you are developing a company that is planning to sell software for preclinical drug discovery or even early clinical, you are very unlikely to make a lot of money because the market is actually pretty small. But it's very, very important to have community of users because you don't really take their data, so you want to protect yourself from their data, but you take their proprietary usage validation data. So they can either like or dislike your models. And if they like the model and you see that it works, you put it everywhere. If they don't like the model, you probably want to replace it with something else or reinforce it. We also do a lot of aging research, it's kind of at our core, but we also started doing some work in sustainable agriculture to understand chemistry much broader, so to understand what is good for certain plants or animals and what is bad for bees and other insects, to understand the differences between species. We also started going into CO2 capture and recycling and other areas that help us get a broader understanding of chemistry and biology. We are truly global. So now our kind of global HQ is in Boston, but most of the AI is done in Montreal, theoretical AI, and Abu Dhabi. That's applied AI and theoretical AI. And we also synthesize, test our molecules and the target hypothesis in labs around Greater China. We have software on the market, so covering biology, chemistry, medicine, and science, general applications for science. Today, I'm going to talk about world models, Precious 1, Precious 2, Precious 3 GPT. That will be pretty cool part of the lecture. So life models, systems that understand biology and time. But the most used system is called PandaOmics. It allows you to identify and prioritize protein targets for a variety of diseases. For example, if you found a bunch of targets that you believe are implicated in aging, you might want to prioritize them for specific diseases, rank them, see which ones are commercially viable, which one are more druggable, which one are safer. So this kind of tasks can be done by PandaOmics. Chemistry 42 allows you to generate molecules with the desired properties. and allows you to very rapidly design molecules with specific drug-like parameters. And this system is currently deployed in many pharmaceutical companies. In clinical, predicts the outcomes of clinical trials, and DORA helps you deploy armies of agents, researchers, so robot researchers that perform certain tasks, and then can write a research paper for you. If you like, I can show you a demo. And utilizing this platform, we very rapidly developed a pipeline of our own therapeutics. So TNIC is our highlight. So the target that we've discovered around 2019, utilizing aging research and artificial intelligence using biological data types. So it's a very new target. Has never been in the clinical trials before. There was no very selective, specifically designed chemical matter for it. We designed our own chemistry, took it to phase 1, phase 2, and now with phase 2A is completed with good safety and tolerability. Plus, we see unexpected efficacy. So that's a huge moment in my life. Some of those drugs are very commercially viable. Well, all of them are supposed to be commercially viable, but some have been proven to be commercially viable because, for example, USP1, we licensed it to a company called Exelixis. They paid us $80 million cash upfront, and then we have milestones and royalties. This one, CAT6, actually also pretty interesting target, which might be implicated in aging, but here it's purpose towards breast cancer, was out-licensed to Menarini, and it's a deal worth over $500 million with $12 million cash upfront. So it's very, very important to understand that we are a factory of drugs. Our job is to discover, develop, and then license them out. We actually don't want to take them all the way to the market ourselves, at least not yet. Maybe, who knows, one day. But we want to establish a very sustainable model where profitability would be one of the very important parameters. And I mentioned DORA, so if you want to utilize this tool, it's actually pretty cool. Let me see if I can show you how it works. You know how ChatGPT works, right? So here the interface would be somewhat similar, right? So imagine that you want to create a new research article for a gene or disease, a research paper, research article. Let's say we look for some target that you know, maybe mTOR, and disease. Let's say we go after maybe let's say chronic kidney fibrosis. Well, let's go chronic kidney fibrosis. Chronic kidney disease, sorry, fibrosis-driven. mTOR, therapeutic target for CKD. So here you can upload your papers that you want to be referenced and read by the system. But let's give it a second. It will quickly tell you what kind of sections it will generate for your paper. You can either remove them or add some of your custom data there. If you have some of your own results or introduction papers, add additional tools. Now you will get to the research plan. And in the research plan that the system will provide, it will already explain what it's going to do, but you can modify it. So give it a second. So now see, in less than a minute, it created a research plan. So it will go and talk to multiple databases about the omics data sets. It will talk to perform network analysis. It will look at core genes. But let's say we also add additional sections and ask it to perform methylation data analysis in the context of disease. And let's say literature review. perform, literature, in the context of CKD, and assess novelty. Okay, so now let's submit and start generating. So now the system will spawn an army of kind of robot researchers. See, it's going to have science reference finder, omics data analysis, web searcher, and it will work with many tools that InSilico has developed. And while I'll be presenting, it will write a paper. So let's go back to the presentation. I hope you can see my main slide. And I'm going to switch gears right now. And I know that I'm a little bit out of time, but I'll try to be very quick and talk about world models. So world models, you might have seen this paper from 2018 by Jürgen Schmidhuber, where he, again, like with many other concepts, he pioneered this. World models were proposed before, but here he actually implemented one. It's a model which understands the basic concepts of the environment, which includes vision, memory, and a controller, and can perform actions within this environment. And they actually put it on GitHub, so you can have a look at it. It's a little bit old nowadays. There are many better models, but definitely cite Schmidhuber with this paper. If you know who Schmidhuber is, it's a very interesting figure. So regardless of how much you cite, you would need to cite more. And other people, of course, picked up and started talking about multimodal foundation world models, where you implement where you learn some of the temporal understanding of the world in order for the model to create a representation of the world from text, for example, or from other parameters. So utilizing generative reinforcement learning. So look at those couple papers, and one of them is proposing that Sora, the one really cool model that allows you to do text to video by OpenAI, very wonderful, popular model, is also a world model. And you probably have seen these, right? So you can see that they can create up to a minute of high quality video from a prompt, with the desired properties, and you can modify it. So you can see that this model understands what's going to come next, right, in terms of the imaging. And that's a very important concept to understand before we go to live models. And they can also, of course, go after continuous loops, and you can really see that it understands objects in time and what's going to come next. So we can do the same thing for biology. So think of a non-text imaging data. Think about biological data types. Can we train deep neural networks, generative systems to understand the different biological concepts in time and simulate life at the level of molecular biology? So I would like to propose life models that are world models that learn basic biology of life in time. So how would we train world models to learn the concept of time in living system? And one feature that is present in pretty much every biological system, with any biological system, and in most non-biological systems, unless you go, sub-quantum, is age, so time. And it's present on pretty much every data set that you are going to touch. This feature was used previously to develop aging clocks, accurate predictors of age using multiple biological data types. And kind of the first biological data type to be super popular in this context was methylation. It wasn't deep learning, but some form of statistical analysis and machine learning performed by Steve Horvath, where he demonstrated in 2012, 2013 that he can use 353 methylation sites to predict age with 2.9 years mean absolute error. Very important to also remember Hanam and Zhang Kang, who performed similar studies at approximately the same time. And around 2016, my group started publishing quite a lot about the applications of deep neural networks for predicting age, for predicting chronological age. And in 2021, we published a small perspective with Dr. Li Kaifu, very famous AI scientist, on how to use deep generative reinforcement learning for all kinds of applications in aging research, starting from predicting of biological age, and referenced many papers that we've published previously. And this is how it would look like. So you collect multiple different biological data types, non-biological data types, epigenetic data types across the entire lifetime, and train deep neural networks to predict age and predict health status. And from there, extract pathways, targets, generate synthetic data. So here we kind of explained the concept of life models. And in 2022, when transformers became very powerful and multimodal generative models became available, we decided to switch from aging clocks that are designed for aging research, can help you extract some aging signals, can help you identify some features that are driving, that are important for the accuracy of the predictor, to life models that can generalize multiple biological processes in time, can do cross-tissue, cross-species analysis, perform in vitro simulations, and are designed for abstract research reasoning, and can perform multiple tasks. So if you are interested in kind of multimodal deep learning models, I highly recommend looking at Gato. So when it was published in May, I think 2022, I was blown away by the capabilities. It's a very important paper by DeepMind. And we started developing systems that are similar, but are focused on, first, prediction of age and generation of synthetic biological data using age as a generation condition, but also can perform multiple tasks. We call it precious, of course, because it's one model to rule them all. Since then, we've published Precious 1 GPT in aging, Precious 2 GPT in NPG Aging, it's an Nature journal, and Precious 3 GPT is currently on archive, and you can use it if you make a significant contribution to this platform or use this system in a non-conventional way. We might actually add you to the paper because we haven't submitted it to a peer-reviewed journal yet. So here is a timeline of those models and QR codes for those models if you are interested in using any of them or looking at the papers. So our first model was transformer plus methylation that also allowed you to do target ID and age prediction. Our Precious 2 GPT is a transformer diffusion, can generate synthetic data and also perform target identification and many other tasks. But Precious 3 GPT, it has multiple different omics, methylation, transcriptomics, proteomics. It has 3 species. It is mouse, monkey, and human, transformer-based, and it also can provide you with the ability to identify geroprotectors. And I'm going to skip a few slides because you can read about the capabilities either online or in the preprint. But I highly encourage you to take this model via open access, either on Hugging Face. It's also available on Discord if you want to play with it. It's instructional. And there are some community members who are very active in aging, like on Tukulaga, who recently, during ARDD, used this model to develop an application, which was really cool. And this model can identify targets for a variety of diseases and also predict age. It can work with multiple species, so mouse, monkey, and human. It can also do zero shot, so it has never seen a dog. It can predict age in a dog or annotate its tissue using methylation transcriptomics, and can also synthesize some differential expression, differential methylation in response to certain compounds, and can perform many other tasks. Again, play around with it on Discord or get it from Hugging Face. We also validated this model using our robotics lab and predicted some compounds that might have geoprotective properties. We haven't tested them in animals yet, but they have been tested in cells. And we also are now working on automating many of the validation tasks for those live models. So you probably know that I have a massive lab, fully automated, that can generate data from every sample, methylation transcriptomics, and several types of imaging data, discover novel targets, identify compounds to repurpose, and constantly train AI models. So I'm not going to go through this. And one paper, if you want to read just one paper about our research so far, just read this one. It was published March 2024, Nature Biotech. Small molecule TNIC inhibitor targets fibrosis in preclinical and clinical models. So in this paper, we actually explained how we used biological data to identify a target. We did hallmarks of aging assessment for this target for the first time, and it's in the paper. Proposit has a timeline of discovery, generated small molecule chemistry for it ourselves, so 79 molecules generated, so it's not a repurposed compound. It's very easy to repurpose, but generating your own chemistry requires guts and significant risk-taking. But then it pays off because it's commercially viable, more commercially viable. And we did indication prioritization using AI, performed many different experiments in vitro, many experiments in vivo, did indication expansion using robotics, took this molecule into phase 0 human clinical trial in Australia in healthy volunteers, then performed 2 phase 1s in New Zealand and in China in healthy volunteers showing safety. And now we have phase 2A with safety and, again, unexpected dose-dependent efficacy. I really hope to publish this data soon. We're currently crunching through the numbers, but it looks extremely promising. And in this paper, so this target, in one of the other papers that we published, we implicated TNIC in many hallmarks of aging. So it's a promising target. I highly recommend looking at this target in the context of multiple age-related diseases and also in the context of aging. And now, as I mentioned, we've got one of the phase 2A trials complete. So far, I don't know of any other AI drug discovery company that managed to go through all of these steps and complete a phase 2A study in a clean way, also with multiple published papers in a very transparent way. So I hope that we succeed. If we do, it's going to be the first of the first of the first. So for both aging research and AI drug discovery. And actually, this team, Cat Longevity, that I met at Longevity Summit Dublin, recently actually showed me that this target might be also supported by methylation aging clocks from Steve Horvath and other labs. And the 3 papers that were published by Horvath and Gladyshev and many others over the past 2 years on multiple mammalian species rank TNIC very highly. So it's just this book is regulated by some of the highest scoring methylation sites in multi-species aging clocks developed by Horvath. So it's kind of additional indirect evidence that it might work in aging. And it also scores very high from Precious 3 GPT in top 20% of all druggable targets. I think that aging research, and this program shows you that aging research is essential for drug discovery. If you want to learn about drug discovery and aging research at scale, come to Aging Research and Drug Discovery meeting. So this year we had our 11th year. We had more than 40 startups sponsor the conference, and very prominent pharmaceutical companies came to the meeting. In 2025, we're going to have our 12th annual. Please register early because this year, almost 2 months in advance, we ran out of tickets, ran out of sponsorships. So this industry is definitely exploding, and the most credible conference that attracts big pharma and investors and very, very highly ranked scientists is likely to be this one. So plan for 5 days in Copenhagen, and just make sure that you don't, go around with flags about immortality or anything like that. It's not about this. This conference is more about utilizing aging research as a platform for drug discovery and understanding human biology. So now I'm completely out of time. I just want to talk about one of my passions, which is longevity architecture. I think that once we get enough resources and build very sustainable business, we should start thinking about how to integrate longevity biotechnology at scale, where in one building, so without the need to travel, you could do AI, biotech, experimental work, clinical work, integrate the hospital facility and the robotics facility, and ensure that this building is also a landmark for the city that it's based in. Here I'm portraying Hong Kong, but it can be built anywhere. So you would combine really prestigious, very expensive real estate with a super high-end hospital focused on non-infectious diseases, biotech ecosystem, and even a small part would be provided to the biotechnology leaders of the biotechnology companies who have pretty much unlimited access to capital anyway, but wouldn't be able to get this ecosystem anywhere else in the world. So that is kind of my vision for the future for the integration of multiple parts of biotechnology. And here I would like to pass the word to Dominika Wilczok, who is my longevity partner and who is going to talk about more frontier technologies in our field. Dominika, take it off.
Thank you so much, Alex. Thank you for the kind introduction. Again, I'm Dominika Wilczok. I'm affiliated with Duke University. And as Alex was talking about the vision of the future, I want to inspire you to perhaps dedicate a little bit of your time to going into more emerging areas of longevity technology. So Alex mentioned the longevity industry, but speaking of this future, we first need to ensure that we actually get there. And you most likely came to Zelar.Citie to get inspired, but we need to acknowledge and somehow take 5 seconds to realize that we are in a bubble. And if we speak about aging every single day, we do realize that we can impact it to quite some extent. But people outside of the bubble, they might have heard of healthy aging, but they don't really know about the full capabilities. To make them know, to make them fully aware of how far we can get with manipulation of aging track, we have to empower and educate physicians. And this is exactly what we do at the Longevity Education Hub. This is the educational hub developed by Alex, by Dr. Evelyn Bishop, who is the primary leader of the hub and one of the most, if not the most inspiring pioneer in longevity medicine. Her Excellency Dr. Mariam Matar, Dr. Morten Schneid-Knudsen, and Dr. Alexei Moskalev. They all created courses in longevity medicine. The first one is the basic introduction into aging research. The second one is the chemistry and biochemistry of aging research and investing in longevity, all translated by real physicians, by volunteers. As much as I love AI here, there's need to be precise in the language terms to all the languages you see below. Right now, we have more than 8,000 learners across 115 countries all around the world, and 93% of the students are physicians. So you can actually imagine the impact that the course has on daily practice. The paper about this is actually going to come out very soon, so stay tuned for that. But the one important thing to say is that you don't have to have an MD degree to get signed up for the course. Just follow the QR code that I will share in a second. And even if you're a high school student, you can still sign up and start learning. The main feat I want to talk about in this part of the presentation is that longevity medicine is now a part of the mandatory MD curriculum right now in Indonesia, which is the first university in the world to ever make the aging research and knowledge of longevity medicine mandatory for students to learn before graduating. That means no MD degree for you if you don't learn about longevity medicine. And this is a milestone, huge milestone in the area, and I'm very proud to say that our hub is the parents of this feat, Dr. Birao with Kansono, just right here, and we have more universities coming in early 2025. Of course, the Evolved wouldn't be possible without the active volunteers, the physicians, the researchers who work every day to make the hub even better for everybody around the world. And if you want to be the part of the team or the part of the students, you can just follow this QR code right here and get signed up for the courses. You'll get a CME accreditation if you're a physician, or a certificate signed by all the authors if you are not a physician. And if you follow us on LinkedIn, you also have access to biweekly newsletter about aging research. But we have to ask ourselves another most important question. In 40 years, we're all patients, just as Alex really likes to say. So what if the cure for aging is not here for our time? What if we get, let's say, terminal cancer before we can impact aging? And thus I want to talk about cryopreservation today, and actually want to argue that if there is even a tiniest voice in you saying, okay, which part of longevity do I want to go in? Or perhaps should I use my skills in a slightly different area, but still connected to aging research? Cryogenics really needs your help. And we actually might have all the theoretical knowledge of it that is needed, and all the technology is available now or in the near term. We just have to combine the pieces of puzzle to make it all work together. So as Alex mentioned before, we talk a lot about this. Our nights are very filled with cryonic discussions, while Alex focuses on AI and biopharma approaches to that. I focus on the emerging tech and BCIs and how to actually combine it all together. So together with Alex, where we were thinking, where are we now with cryo? What would we do if something happens? And where do we actually want to go with that? We figured that there are 3 levels we want to climb through, we as the field of cryonics. We are right now at level 1. That means that cryopreservation of the whole organism is already possible. There are 5 companies around the world that do that. One, the closest to Berlin, is actually in Switzerland, tomorrow Biostasis. However, rewarming is right now not yet achieved. Not in
I
a
just
human,
want to
not in
interrupt here for a second. So we're not yet at level 1. So level 1 implies that we actually do have the capability to reworm in the future. So it goes through all the processes that gives us a lot of confidence that with today's technology, we'll be able to do that. So currently we're below level 1. I think currently with the current technologies, we are not yet able to say with high confidence that people who are preserved using today's pretty dramatic technologies, we would be able to rewarm. So we are actually thinking a lot about how to develop the optimal experiment in order to prove that to gain confidence that rewarming would be possible. So we are discussing and supporting certain academic labs and inspiring some of the academic labs to switch into this type of research, specifically focusing on technologies that don't require cryoprotectants.
Exactly.
Yeah, sorry, just Dominika made
no, it's very
Sorry.
Thank you so much, Alex. It was a mental shortcut, but yes, there are companies in the market that offer you cryopreservation opportunities, whether that's at any possibility of rewarming. That, as Alex mentioned, leaves a lot of questions. However, the second level we actually want to really get to is to, with full confidence, to cryopreserve and then rewarm. Level 3, which is not the subject of today's presentation, but rather future outlooks, is that we could actually manipulate the preserved organism without breaking the cryopreservation process. So right now, to understand the biggest challenges of what actually prevents us from getting into level 1, we have to understand that cell damage and apoptosis is the main blockage here, and this is mostly due to 3 issues. Cryoprotectants are substances that prevent ice crystal formation. Ice crystals are hard or sharp. They actually break the cell or cause osmotic stress that makes the cell die in the end due to damage. And ischemia, so not enough blood flow in an organ or a part of the body, also is a huge parameter that needs to be optimized. So to fully understand that, we actually need to see how the crystal formation forms inside of a tissue. So normally a cell hangs around about here on the y-axis at the 36 degrees centigrade. If we want to cool it down, we actually need to somehow pass this ice formation phase from the liquid phase straight into the glass phase. And that is achieved right now through vitrification or isochoric cryopreservation, which I will talk about in a second. But basically what you need to understand that we need to very rapidly cool it from here to here, not to prevent ice crystal formation. How to prevent ice crystal formation from reheating, so going from here to here, that's another big question. So just to reiterate, a cell that undergoes cryopreservation is exposed to a lot of dangers, cryoprotectant toxicity being one of them, and then tissue damage due to ice formation, and ice formation results is associated. So what can we do right now for cryopreservation? We can do single cells, sperm, egg cells. These are all possibly cryopreserved and widely used in IVF. But you need to understand that the traditional cryopreservation, quote unquote, is called vitrification. Here you can see the image of the kidney, and although we cannot rewarm a kidney to full functionality yet, I think this picture really nicely exemplifies that vitrification is actually getting the biomaterial into a solid glass-like state. It requires the high use of cryoprotectants, so for example, glycerol or trehalose here, and rapid cooling with liquid nitrogen. There are other experimental methods in that. So given that cryoprotectants are actually highly toxic in high concentrations, to lower their concentration, there is a method called isochoric cryopreservation. That means that you keep the volume constant and therefore create high pressure and create cryopreservation from that. So one thing that we actually need to understand, and even though it sounds nice to say let's freeze people, cryopreservation does not equal freezing, because slow freezing leads to crystal formation, while the other 2 methods rather don't. So as I mentioned before, cryogenics is a multi-parameter optimization problem, because there are so many things we need to control, from pressure, from temperature to concentrations and gradients. So one thing that you might recall from very early days in your education is that noble gases are actually very good at multi-parameter properties. It's because they have low chemical reactivity, low thermal conductivity. That means they wouldn't form bonds with any of the molecules, and would allow for uniform cooling and rewarming, also due to high pressure that induction of these gases would create. So we talked about cooling, but what about rewarming? And as I mentioned, only single cells are possible right now, but something I want to draw your attention to is actually this patent filed by Professor Dao-Yong Ga from University of Washington and his team this August this year, about using single-mode electromagnetic resonance for rewarming. They used a rabbit vein from the neck. But before I go into how they actually did that, we need to understand one more theoretical concept, which is the thermal runaway problem. So basically, if you imagine there is your biomaterial and you heat it with electromagnetic radiation, surface will heat up first. And it works like this, that the hotter surface just absorbs even more electromagnetic radiation, and the inside, the cooler parts still stay cool, which creates a gradient and thermal stress and tissue damage. However, when we use just the single mode of electromagnetic radiation and combine it with superparamagnetic particles such as iron oxide, they evenly distribute the radiation, the heat, and therefore there is no gradient and thermal stress. So this was achieved and patented using rabbit veins from the neck. And here you can see the graph of the mechanism that was used. Basically, the biomaterial, the vein, was placed in a container. That the container was placed in the liquid with cryoprotectants and the superparamagnetic particles, and that was still placed in another container and generated electromagnetic waves for rewarming. And the magnificent effects, we can actually see it here, that compared to standard rewarming methods, which is just a bath of water with 37 degrees Celsius, their method actually achieves statistical significance, and viability is almost as high as the control, which was not cryopreserved. And you can also see amazing difference between standard methods and their method in vein response. Same around here, the recovery rate, you can see that this is just standard control, electromagnetic radiation, and preventing the thermal runaway with magnetic particles. And same goes for cell metabolic activity. You can see it almost goes to 100%. So the next step, just like Alex mentioned, we need to get a successful rewarming of impulse. And if there is any postdoc in the audience which has any experience in cryonics, Professor Alana Spurugudzik, a huge leader and mentor in the area of drug discovery and chemistry, is right now looking for a postdoc in exactly this area. So you can just follow the QR code below and perhaps contribute to one of the biggest feats of aging research before we get the cure. Thank you so much for your attention. If we have any time, Alex and I are happy to take any questions.
Yeah, here I just wanted to make a comment. So Alana Spurugudzik is one of the one of the pioneers in generative AI for drug discovery. So in 2016, we used to compete. Now we have partnered on multiple projects, including some applications of quantum computing for drug discovery, and recently synthesized the first molecule generated on a quantum computer on IBM 6-qubit and tested. Alan's group is probably one of the best equipped to go after fundamental science here. So, and he'll definitely be exploring multi-parameter optimization problems with real experiments. So please do promote that postdoc position. And if you're really good in physics and preferably also in AI and willing to work 24/7, that's your ultimate job. Because at the end of the day, if you think about, what would happen if we get stage 4 colorectal, right? Or you get prostate cancer metastasized in the bone, or some other form of really rapidly progressing deadly disease. There are not that many options, right? And you wouldn't have a lot of time to wait for the clinical trials to complete. And the idea is to create a really sustainable business model around this technology in order to give patients time to wait until clinical trials come out and read out, and maybe they would be able to get a cure they need. It's not, for preserving dead people. So the idea is to come up with the technology that allows you to wait until clinical trials are complete. Of course, there are many business opportunities in preserving food, in preserving organs. I think that a lot of companies are going after organs, and they go down the rabbit hole of utilizing very toxic, well, non-toxic or whatever cryoprotectants. It's not the ultimate solution, in my opinion, right? If you want to industrialize that, you need to go minimum cryoreagent focused technology. And I think Dominika is exploring many technologies right now, talking to many labs. So please talk to her if you're interested in this area of research. If I were to start a company today, I would actually focus on that, because that's probably the industry that is going to be bigger than generative AI by far. Because at the end of the day, all of us would want to see more life, And if I had the capacity, I would focus on that. But fortunately, I've gotten silica, and I'm not Elon. I'm not gonna do anything to jeopardize in silica at this moment, because it's the most precious golden goose that is laying golden eggs, also in the area of longevity. But if you are interested in going into cryo, I would love to have Dominika support you in some way.
Exactly. You
Thanks a
can
lot.
reach it, both of us very easily on LinkedIn. So just don't waste time and go into it.
Alex, Dominika, thanks a lot. Please, round of applause to our speaker. I have so many questions. Some of them you actually answered in advance, like the last, what company would you do if not in silico? But I cannot usurp the microphone when the audience have so many questions. Please.
On the Cryonics front, one solution to the kind of thermal runaway problem is to basically design like a thermochrome, like a particle that changes how the light is like transferred into heat based on temperature. And these exist like in material science papers. I found some, but there's actually like not. This is like a really good optimization problem. Like if someone actually were to use AI to like design the like molecule here, that would be where I would start. There's something that kind of like switches conformations once it gets like above like 0 degrees, and then you like never have the problem of like overheating.
So again, what kind of particle do you want to design?
A thermochrome, so something that changes how light is absorbed based on temperature. They're like, you know, these like packs that have like kids use them, like they change color when it gets hotter. And you can fine tune them quite precisely. So yeah, the idea here would be that you have something that basically becomes transparent once it gets too hot. So that region of the biological tissue doesn't heat up further.
That would be very interesting kind of direction. I guess currently you would be using ferromagnetic nanoparticles for kind of rewarming. So is this what you want to achieve, to basically for rewarming to use a particle that would help with reheating?
Any other questions? Lawrence?
I'm curious how you get to be so productive, and what your advice is regarding the balance between, you know, working a lot because this is life and death, and then kind of also taking care of your own health and longevity.
I am extremely unproductive. So I wish I could be more productive. Like look at what Elon is doing, right, on his industries. We should all learn from him. I think that from my experience, it's very important to just focus on your kind of main idea, main business, and ensure that you do whatever it takes to push it through, right? So it's very easy to be distracted by new shiny things, but it's also very destructive when you get distracted. And once you selected your kind of path, it's very important to commit to it, especially if you are going after a certain drug. So if you are doing drug discovery and you committed to a certain disease hypothesis or a certain protein target or a set of targets, it's just very important to commit and just don't steer away from it, right? So I've seen so many brilliant minds with, you know, PhDs from Harvard, MIT, Stanford, etc., and 10 years you know, under the belt in pharma and biopharma, abandon extremely promising programs in big pharmaceutical companies or in small biotechs because they decided to chase some new shiny things, new shiny thing, you know, that helped them advance their career or just because of curiosity. If you are going after longevity biotechnology, I think you need to be prepared to play a very long game and just optimize the entire life for that particular goal. So my life is optimized to make in silico successful, because I think that if in silico is successful, it will help the rest of the longevity biotechnology industry, because again, everything else I've seen so far has failed or has not failed yet, but it's very likely to fail. But I really want to be committed to that particular goal and mission, and even personal relationships. So I've never met anybody more capable than Dominika, so we're very happy to be together, and now we're working together, and we're complementing each other. So maybe one advice is that find yourself a longevity research partner who is smarter than you and can help you perform, also understands why you are doing this and why you're putting so much effort into this and not something else. Because a lot of people are talking about work-life balance, but if you start talking about life cancer balance or life aging balance, those things don't take vacations. So you need to just focus on what you are committed to, and until you either succeed or fail.
Exactly. And to come back to your original question just a little bit, work-life balance is almost as much of a hype as the AI right now. Everybody talks about it, and there is a nice parallel between the work-life balance and longevity medicine, actually, which is to understand that we're all individuals. So the studies of N equals 1. A lot of people will try to tell you a lot of things on the internet to follow this rule, to use Pomodoro technique, to use everything. But the most important part is to, just like Alex mentioned, find your purpose, find people who understand why you are committing that much time and efforts to that purpose, and just don't get fooled by all the trends that try to fool you into just sticking to one method or another. Just believe that you are going for a goal. And even if you're going to fail, even if you're going to succeed, you're still going to get a lot of new ideas and new lessons on the progress to that.
Very wise words from longevity, Rick and Morty. We can squeeze in one. We can squeeze in one or two questions. Victoria?
Thank you, Alex and Dominika. I wanted to ask you, what's an underappreciated area in longevity biotech nowadays?
There are so many I would have to thank. So I think that if you put cryo into the longevity biotech kind of bucket, I would say that's probably the most unappreciated and will be the greatest industry of our lifetimes. If you are talking about kind of basic research in understanding of human biology of aging, I think that kind of cross-species organ and cross-species transgenic experiments are probably the most underappreciated. So imagine if you were to modify an organism that is very similar to us to produce high quality organs that are very compatible. You could solve a very substantial problem. Now people are talking about brainless clones, etc., and that's a great area to explore as basic science. But as it progresses, the society would probably not allow this technology to exist at scale. But transgenic experiments with non-human with animals are likely to be very productive. And you've seen that Craig Venter and Martin Rothblatt and many others started investing in this, but so far this is just scratching the surface. So somebody needs to just invest a lot of money, a lot of effort into this area and deliver a working, biocompatible kidney, for example. That would change the world, something that, you can put into yourself with high confidence. And so far, I don't see many groups working on that.
And to add to that just a little bit is that we really do need more clinical trials, even if they are just mentioned on ARDD this year. Very great presentation about N=1 trials. We really do need more human clinical data on supplements or everything. We just need validation.
Yeah, I also want to use this opportunity to promote week 4, or week 5, which will focus on cryonics and other future technologies which Dominika described. Emil, one of the founders of the Dominika mentioned, will come and talk as well. It will be possible to engage in these really cool, interesting talks. So, any one last questions? Do we have it? Okay, then I will intervene and ask Dominika. What influenced your choice to focus on longevity when you was so relatively young? We need more people focusing on longevity at a very young age. How to do that, in your opinion?
Oh, well, thank you for asking this question. My story actually started with Alzheimer's disease, but not because I have any family history, but simply because when I was transitioning from junior high school to high school, I just found myself with a lot of time because school wasn't enough. I just, found that classes were not enough, extracurriculars were not enough. And I just needed to do something with impact. So as I was a bio nerd from the very, very early beginning, I was just reading through everything about medicine, about biology, human biology at the time, and I just noticed the pattern between in the Polish medical news forums about Alzheimer's. Every single day there was a news about Alzheimer's, about either a new potential therapy or a therapy that failed, or there was just so much inconsistency. So as I identified this pattern, I decided to get into that, and I found that there is so much misinformation at that point, and just we don't still know what actually causes it. And I decided to start a social project back then about how correctly identifying Alzheimer's back then with AI, just making people aware that there are opportunities for that and what can we do with that in the future. So as to maybe generalize this more from my case to something more general, if you are parents, if you have young people around you, just identify that classes, especially now with large language models, that are just not enough. The young people can explore every single thing they want in the universe just by asking ChatGPT questions, ChatGPT with infinite patience and creativity. So just see what area of science or just life they lean into it, try to identify the interest, and just don't be afraid to dive deeply into the topic, immerse yourself, and don't feel the narrative that you are just a student and you need to stay just in the theoretical world. Just use resources, reach out to people. People are really willing to help young people get into any areas of their specialty. So just put yourself out there and you will never get more than just immersing yourself in person. So use ChatGPT, learn a lot, and then try to innovate based on the knowledge you gained, all by putting yourself out there. Don't keep it in your drawer.
Yeah, thank you. Thank you for going over time and answering questions for us. And thanks a lot for investing this time for us. I hope it will help to start new companies and help existing companies to move forward. Thank you very much. You're doing great work for longevity. Looking forward to seeing you in person and have a nice rest of the day.
Thank you, Alexei.